to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 67th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Moreno, and I've been in the LBA and cell and gene therapy space for over 29 years now. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for over 28 years.
John and I are both subject matter experts here at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas, John is in upstate New York, and our producer is in Missouri, so we're all over the map a little bit. We are ecstatic to have you listening to the 67th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a single listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing, before discussing any feedback you may have had for us. We're constantly looking for topics, and we'd be happy to discuss something that you may want us to cover. So again, we're ecstatic to have you here, and we are looking forward to a fun episode. To kick us off for podcast number 67, John is going to go over today's podcast topics a bit before we jump into the news and resources section. John?
Yeah, as usual, we'll touch on the latest medical research news with a few items. Then we're actually going to have an interim discussion of planned attendance and presentations at upcoming trade shows, because that's something that's of big and real importance to us as we move forward and try and connect with our peers in the industry. And the main topic today is evolution of drug and PK modalities and the impact on regulations. So really talking about complexity of new drug entities and how that's impacted how we work and how we measure, how we look at PK and the regulations to meet that.
Yeah, John, almost like a three-parter today is what we're going to go for. Yeah,
Exactly.
Mixing it up a little bit. Crazy here on a Friday. But no, John, with that, thanks for getting us teed up for today's podcast. And why don't we just jump into the latest news and resources? John, what do we got for?
Sure. First up, I like the approach behind all this story. The headline is "Scientists Studying Bears Find Path to Treat Deadly Blood Clots," and it's a group led by Manuela Thinel, a physician and researcher at Ludwig Maximilians University Munich in Germany. Looked at hibernating bears to see how they could possibly hold an answer to thromboembolism, which is a big problem for people who are, obviously idle in bed in hospital and things like that, where it can lead to clotting and then complications from that. And I think on a personal note, my father's mother died very young, and he was convinced that she'd spent some time in hospital, and this was at the root of her early demise. So to find someone looking into this is really, really good. And it's like I say, it's the problem where humans, if they're immobile for weeks or months, they're at high risk for potentially fatal blood clots, and that's the issue here. But in talking to colleagues and then researching further, they're thinking bears do that every year, and other animals, and they suffer no blood congealing consequences or any other physical debilitation. I mean, you know when you watch like TV shows and you see people who are bedridden for ages, but they get up and they're immediately running around. It's like, well, that isn't real. You're gonna have muscle weakness and all that. Well, bears have licked that problem.
That is so true. I don't know if you had anything more to add. I was going
to go ahead and talk, and
it's like, it reminds me of like Newton having the apple hit his head, right? That's what this is.
Yep.
By just observing, thinking about it. These scientists were, I think it's Thiel there. They're looking at a bear, thinking, how are they hibernating, right? And what a great, I think of it as reverse engineering to solve the problem. So,
sure. Yeah,
And then I mean, diving into it a little bit, the heat shock protein, right? Heat shock protein 47 appears to be a big part of what they ended up kind of figuring out was part of it, right? They looked at spinal cord patients as well, so it led to a whole series of. This is quite a long article here, right?
sure. Oh,
It
absolutely.
led to a whole series of events through them trying to creatively, not just bears. Again, it was that spinal cord and one other disease. And go ahead.
They say basically pigs, mice, spinal cord patients, and then people who have done like research for space,
Yeah,
what to simulate physiologic effects of space flight. And you're right, it's HSP47 heat shock proteins found on platelets, and basically platelets are there to help patch wounds and stop bleeding. It's the everyone every species that they looked at has this that's HSP47, and also in looking at when they see lower levels of HSP47 in humans, they found that clotting was actually reduced as well. So there's a number of things pointing at that.
yeah.
Um,
It's just great. Neutrophils are a player involved here, and they talk about um, neutrophil extracellular traps or NETs, right? But
sure. Yep.
so it's really I don't know. So there is there a therapy in here, John? That I guess
No,
before
no, no.
as we start to
This is
move on, this is
the emphasis. This is all early research.
yeah,
It's like, you know, having looked at this, we may find that it's ultimately here's what we've identified as playing a major role in this, and it's this HSP47. So ultimately, the next step is then, well, what medications can we do to, or can we develop to suppress HSP47 and thus the risk of clotting. So it's a really early identify a potential agent that's an issue, and then obviously there's a lot of work to to come
yeah.
to possibly come up with something that can fix this. But I mean, it affects it can kill up to 100,000 Americans every year. So it's not like this is a minor problem.
No, it's going to
But
go up too, John. Right? I mean, this is no different than sitting all day, right?
Mhm.
This is part of it as well. We're certainly a generation, ourselves included, that we're not we're not up and about as much as the previous generations were, and I think the ones behind us are even going to be more sedentary. So, like, yeah, this is definitely something that has a lot of future potential to help solve other ailments. Right? This
Yeah.
is wonderful.
Yeah.
It's
It was a long article, but the actual it's easy to distill what is the high points out of it. But the like you say, they actually as part of it, I love this that they actually go and sample blood from hibernating bears,
yeah.
and so they'll be back out. I think they're talking going back out this winter or this past winter, for having identified the protein. Like you say, it's a biomarker, absolutely. Having identified the biomarker, now trying to figure out the mechanism, and that's where the mechanism how it functions, and that's going to then hopefully play into development of medications. But I love the fact that when they've sampled the bears don't really react. They just sort of like it's like if you're shaking awake when you've been asleep for a long time. They sort of get up, look at you groggily, and then wander off and go to sleep somewhere else. And they're really not that fussed by it all. So yeah, those groggy bears could actually be a big deal for us in the long term.
You couldn't pay me enough to go up to a bear and draw blood from it.
No,
It's
no,
not something I would do.
I'll happily leave that to someone else.
Yeah, no, it's a good way to end a very good story, John. Right? This is
Sure, and
really cool
I
stuff.
think this is it. It's all about new things that I mean they could have a long term effect. So the next
Yeah,
article is in search of new way to deliver gene editors, CRISPR pioneers turns to molecular syringes, and yet again it's the natural world that's inspiring scientists. And I love this. I love the way that someone looks at hibernating bears and thinks, "Aha! I see some of the issues that they can possibly why are they not seeing the same issue when they're sleeping that
yeah,
humans do?" And then this is actually the trigger for this is soil bacteria. And it's some soil bacteria have evolved tiny but deadly injection systems that attach to insect cells, perforate them, and release toxin inside, killing a bug in just a few days' time. And so, but where the CRISPR people are coming from, they're thinking that's a system to inject or deliver medicines. And so with CRISPR, part of the issue is is the delivery system. How do we get what we want into the body, to the body? And the note was made that, we do talk a lot about liquid nanoparticles, but that really directs things to the liver. They're looking for a delivery mechanism that can be more universal than that. And so they basically designed syringes based on what these bacteria do. And then they sort of used it to load programs, but they had to go through hundreds of it, 100 or so iterations before they had a structure that would work. And then it was then, and this is where AI really came into the fore to really help them develop quickly as to reject or see what looked promising to move forward. And they're working with AlphaFold, which can predict protein structure, and then with that they could figure out how to engineer the actual delivery syringe to then get the protein into the or to start thinking about where the protein can be targeted. So I'll pass that back to you.
Yeah, it's John. Like you said, and I'll go back to the Newton and his apple. Right, this is unbelievable there. And I did when I got my master's degree at UMass Lowell, they were big into soil bacteria, and I often some of what I learned there is really been helpful. That at the time I thought, I don't really know why I'm studying this, but it actually this is part of it, right? You learn a lot of different things, and it helps to translate into many things. So a fascinating story about just how the soil bacteria work, obviously, and then just the way they went about trying, John, like so many people, we have a lot of young people in the organization. We mentor here. It's all of that. You said it best: hundreds of iterations of things before it worked. Right?
Yeah, sure.
That's the
Yep.
tenacity that a scientist needs to have, the mindset, right? And then they, AI, right? That's another
Yep.
an advancement, and one we're using actively here at KCAS to help us improve the quality of our work. So that's another, like, all of it's coming together. It's not just, I'm going to go in and try multiple iterations of the assay. It's more, hey, how do I get creative, and how do I think differently? And so,
Sure. Yeah. So,
and
I
then
mean,
obviously this overcomes some major obstacles, and you touched on it with, liquid nanoparticles go to the liver, and then you can't like I think of it as how Wolverine got adamantium in the X-Men series. Maybe you don't know the movie, but they did micro injections across his entire body, but we can't do that either, right, John? So there's a
sure.
systemic
Yep.
delivery I think about, and what a clever little way, and you can maybe add more about that in terms of the Caspase-9, or
Yeah,
how they were looking at it.
I'll just mention. I mean, this is published in Nature, and it's Feng Zhang, who's at MIT, and it was his lab. And like you said, hundreds of iterations, 3 to 4 years to get to this point where they have a potential moving forward. So again, it's another early piece of research that shows the potential direction. It's not ready. It's not ready yet for for people, but they have looked at getting it into the brains of live mice, so that gets around the
Yeah.
ability to target different organs than what we're talking with liquid
Yeah, the
nanoparticle, etc.
blood-brain barrier. The blood-brain barrier has always been a fascinating thing to study, John. Right?
Yes.
Um, your themes here for articles are great, right? I mean, we can move on to the next one if you'd
It's a
like.
new one, yeah. And
Yeah.
I will actually very briefly touch on what I was going to talk about, but then I found this and I thought it's actually made much more sense. But there was a few articles. Partly the reason why I dropped the other one was because I couldn't get to access the original Nature article I'd read about it, which was people analysed Beethoven's hair and
Oh,
think they have identified at least some of the cause of his death, not the reason why he went deaf. That they couldn't do, but they actually had like 8 different samples of hair, 5 of which, based on the timing and characteristics, they thought was from Beethoven, and there was one that had been like a famous lock of hair that someone had got, that they turned out they said that was absolutely not from Beethoven, and
oh,
that had pointed to some medical condition that wasn't the case. But it turns out he was actually prone to he had genes that made him prone to like liver disease, and but at the end of his life he was a bit of a heavy drinker, which I can sympathise with, and that was probably what ultimately led to his sickness and death. But if you get a chance to check that out, that's really interesting, because again it's like having an old hair sample from the early 1800s and being able to from the DNA, and they didn't have a full set of DNA. There'd been sufficient decay that they couldn't do everything.
It's all right, John. Our grandchildren will be using his hair to clone people on their printer using AI.
So I thought that was an interesting aside. I won't dwell any further on that. But this one, this is you'll see the key word in this. But basically, new test detects Parkinson's biomarker before movement symptoms, and again as we're going with an older population, Parkinson's is an increasing issue. It's a debilitating disease, and one of the things that people would like to be able to do is detect it before symptoms appear. So it gives that possibility for early intervention and hopefully control the symptoms, etc. And again, this is another long study published in the Lancet Neurology, and it's a team of scientists with Parkinson's Progression Markers Initiative, backed by Michael J. Fox's Parkinson's Research nonprofit, developed an assay to detect deposits of alpha-synuclein in this cerebral spinal fluids of hundreds of at-risk and early-stage patients. The test, recently commercialized by Amprion as Syntap, was able to distinguish them from non-Parkinson's patients 88% of the time and had a specificity of 96%. Now, it does depend on certain types of Parkinson's or symptoms. I think for a large proportion of Parkinson's patients, they have a loss of smell, and for that group of people, the test was actually incredibly accurate, and I think it could pick up those very quickly. There's people who had a mutation in the gene LRRK2, it was less successful for those, but even still, 68% of people it could detect as having Parkinson's. So, the reason why I touched on that is obviously because we have the SMCX Pro as our sensitive instrument, where alpha-synuclein might be one of those analytes that we can analyze as our biomarker, that we can analyze and provide a similar kind of service.
It's funny, you read my mind, John. We've done some development on that marker and looking at the phosphorylated version of it and the non-phosphorylated version of it on the SMCX Pro, and had some nice successes there. Unfortunately, the company that was researching it, as sometimes happens, they go out of money. I forget. I feel like some of the intellectual property went somewhere, so maybe we can re-engage them. But this is, we're very familiar with alpha-synuclein, and it is it's definitely a critical biomarker in Parkinson's. I think some of this is now John. I think this is obviously diagnosing something is really a best first step towards trying to cure it. But this Parkinson's space is very tough to cure and treat because of just the, we don't understand a lot of pathways that are impacted by this. It's hard to study, right? What models are you using? How do I do it on you? It's not like dermatitis where, it's right on your skin, right? Versus something that's, and I touched on the blood-brain barrier, right? That was a big part of what happens here. And then there's all these really, I think even from, we're a bioanalytical discussion here, right? So bioanalytically, some of the questions, and I don't do this often I don't want to poke holes in something, John, but I feel like there's a lot of complexes of this, and maybe they, addressed it in the way they did it, but it's a tricky molecule to measure, although the CNF they did it in CSF, right? Is that?
Yeah, absolutely.
Yeah, that's definitely a matrix that you have less of that.
I think it's worth looking at further, because they talk about the SynTap, which is a seed amplification assay, but it's actually used routinely in diagnosis of other neurodegenerative prion diseases like
Oh,
Creutzfeldt-Jakob
wow.
disease,
I see.
where it's their use in diagnosing brain pathologies involving misfolded proteins. In Parkinson's, misfolded alpha-synuclein accumulates in the brain as part of what is known as Lewy bodies, which leads to the loss of neurons that
Mm.
signal using the neurotransmitter dopamine. I'm reading this. It's just not very so far away from my area of
Yeah,
expertise.
no, it's
I think
all making sense.
that's why this was interesting, because again it's building on something that is more universally accepted and saying, can we apply this to another disease that obviously we have a need to diagnose, and based on what they've reported on a long study with over 1,100 patients, they feel that they have a
Yeah.
solid path forward with this. So
And
that's why I mentioned it.
yeah, and well, John, like anything, right? Like one of the first things I learned about immunology is it's all your cells are like dual-edged swords, John, right?
Hmm.
If they're overactive, that leads to a disease state. If they're underactive, that leads to you being susceptible. That's no different here. So alpha-synuclein has a function. It's just when it starts to deposit,
Sure.
that's when
Yeah.
it becomes a disease state. So how do you treat that without removing all the alpha synuclein? That sounds dangerous,
Sure.
right? So,
Absolutely. Yeah.
so that becomes like, and then okay, can I? It's not, we you're able to do. We get plaque buildup in our arteries, and they can go in there and kind of clear them things up or build a new one for you. Right? We got great technologies, and the heart can take it. But John, you touch someone's brain, and that's a lobotomy, right?
Sure.
So that so
Yeah.
that that's that's part of the challenge with this. But it is it's great to see. Certainly, it's refreshing. I think we're kind of past the whole COVID, no COVID thing, but these are the types of things that are starting to really pick up in our world,
Sure.
John. Right?
I suppose part of it would be, I mean, that they say that it's the misfolded alpha synuclein that is accumulating that is causing the issues. So it's a question of what is the pathway to the misfolding, and can
So
that
you
be?
think the
I
well,
don't know. I mean, I'm
yeah,
just
no, but I there's another. So most misfolding is a chronic state, John.
yeah.
So acute, right? Acute is something that's acute can cause a spike in something, and then it goes away and you're fine. If
Hmm.
that acute goes to a chronic state, then that's when your proteins start to misfold.
Yep.
But anyhow, it's something that there's some really companies in this space that we know well, John. Right? So
Sure.
stay
Absolutely.
tuned there, and
Yep. Yep.
certainly something to maybe revisit just with our team to see if it's something like you mentioned. We believe that we can measure it. I think we've done it by hybrid mass spec as well, or at least we dabbled in it there as
Yeah,
well.
I'm not sure on that
Well,
one,
John,
but Don could tell you more on that, I'm sure.
yeah.
Yes.
So
So
you got another event before we dive into the main portion here? Is there
yeah,
one
the
more?
other bit of news was really talking about its conference season, and thought we quickly touch on planned attendance and presentations at upcoming trade shows. Um, I haven't got anything planned at the moment, but actually that's not quite fair. I will be on a plane on Sunday, ready to attend the a PBSS meeting in San Francisco, and I should know what PBSS is.
Yeah, it's oh boy, I used to know. It's the old Keiko, which was California, but yeah, it's
I've just been
it's a
on the website too.
yeah. Sorry, I'm not able to help you out either with what it is. Although John, I think I've attended maybe a dozen PBS conferences in my time here. At one point, I was going to two a year. They're
So it's
they're great half day events, right? I
yeah,
think it's a
sure.
half day event.
So it's Pharmaceutical and Bioscience Society, and it's in this one's in the Bay Area, it's in Foster City, California. They have a main topic, but ultimately there's you do have the ability to have a booth and then talk to people, and it's always a good chance to mix with peers, etc. So this is the topic this time is preclinical development IND filing for both small molecules and antibody based
Excellent.
therapeutics. So yes, so I'll be joining Chris Frankovic and spending that on Monday, and then hopefully go and see some customers. I think from there we also you're also on the road next week.
Yeah, I travel Sunday, and I head to Philadelphia for 3 days to see to attend the American Association of Pharmaceutical Scientists National Biotech Conference, or AAPS NBC. There's heavily focus on biologics there. Obviously, it's biotech, and so then there's a very big portion of it dedicated to um, cell and gene therapies. A lot of good. We'll have a panel discussion related to qPCR and digital drop PCR support of this in this space. As well as Don Duffield that's presented by Carrie Vidahal in our cell and gene therapy division, and she's excited to do that. And she's going to be moderating the panel on that. There's about 6 people on the panel from CRO space, and as well as the pharma and biotech space. Dawn Dufield's attending it with me. She's always an exciting and electric person in this space, John, to be attending these things. But I don't know. This is maybe I guess they did away with this conference, but I think this is my 6th or 7th AAPS NBC that I've attended. Probably somewhere, maybe it's 5 or 6, but they're always very enjoyable, heavy in the flow cytometry and ELISpot talks as well, and really excited to go to that. And then there's a heavy immunogenicity track, which is always a hot topic in the cell and gene therapy space as well. Then we got what?
So Carrie's busy this year, so she's presenting at the Spring Focus Workshop, the EBF Spring Focus Workshop on oligonucleotides and peptides. It's a 2-day workshop in Malaga, Spain. So like I say, that's in early June. So that will be. Looking at the program, it will be fascinating, particularly for an LCMS perspective on the.
Are you going to that, John? Right?
I'm not, but I'd like to. So
we can talk about that. We don't have to discuss it on our podcast here.
Exactly. Yes. So yeah, Carrie's presenting at that. It looked, when we had abstracts, we just didn't have something that really was a good fit with the workshop from an LCMS perspective, because I'd already presented the Lego stuff at EBF back in November.
Yeah, she's presenting on aptamers, right? Really good talk there, which is
by flow.
Thing.
So completely different approach, but I think it's great to get.
And then I think we're going to have another podcast between now and then, but WRIB is coming up, right? That's another one that's in June.
Yeah, so WRIB, that's also in June, isn't it? That's Orlando. Yeah. And I think that's more mid to late June.
Yeah, I think it's late June. I forget I know the abstract's actually due today. I believe we have 3 that are submitted already, or at least that I know of. And I think Cyto's coming up. When's the Cyto?
So from WRIB, I mean, at least in terms of the abstracts, I know that Brian Parmentier has one on direct measurement of a small molecule biomarker, and when I say small molecule, it's very small, and that's the challenge of it. It's a molecular weight of around 60, and usually, measurements are more indirect approaches, where they came up with an approach that is a direct measurement by LCMS. I hope that's accepted, because that, to me, that is a really fascinating. Once he described what they'd done, that is a fascinating topic for LCMS, because our challenges, the middle piece is always easier in terms of small molecules, peptides, etc. But the challenge are the very low molecular weight and then higher molecular weight. So to have a paper on low molecular weight is unusual, and that's like I say, that's part of the excitement from someone.
I can tell you perked up. You were all into it. Yeah. I fell asleep, but I was like, no, I'm just kidding.
I'm hoping that's, to me, that's the kind of topic that's translatable to other meetings. And
I don't know. I know we have a couple. Sorry, go ahead.
I may twist Brian's arm and steal the date to do something at EBF later in the year.
No, absolutely. I believe we've got a couple others submitted. I don't have as much knowledge on that, but if they get accepted, I'll certainly know about them. And then, there's another meeting. It's Cyto 2023. That's a cytometry meeting. That's in Quebec this year, May 20th through the 24th. And we do have a couple posters that got accepted there. Of course, trying to remember specifically what they're on, but
Is that
maybe
is that from
maybe what's that now?
is that from both Olathe and Ship,
No, they're
or
both
what?
at they're both from our ship site.
So
I
Ship
feel
being
like
our Springhouse, Pennsylvania
thank you.
site.
Yes,
Yeah.
they're both from there. I guess I could quickly look them up, but I don't. They're anyhow. That's one we'll be at. I believe it coincides May 20th through 24th, like I said, and then I think that covers it for the first half of or at least the rest of the year, John. I don't know if you have
First
any
half of the year,
others.
because obviously latter half of the year we'll have AAPS Farmside 360 in October, and then EBF in November, and yes,
An
that's it.
immunogenicity summit, I believe, as well as an immunogenicity of gene therapeutics is the other. I feel like
And,
we're going
and,
to be
and I
at.
have I know of at least one other meeting that might be worth looking at, but it's too early to say
Oh
anything
yeah,
about that.
I think we're going to APA. I don't know when that is. The American Association of Pharmaceutical Scientists in Boston every year, John. If you're familiar
Okay,
with that one,
okay.
I
I've never
believe
been to it.
Don is talking at that one. Um, but anyhow, that's American. How anyway? Well, John, that was a really cool little portion of our main topic today. um, but maybe we'll transition and spend a few minutes discussing the
Yeah,
main portion of today's talk, and that's the evolution of drug and PK modalities and the impacts on regulations. And John, I'll just kind of
steam right in, and I'll contribute as necessary.
sure, yeah. I mean, what I had vision wise for this was just, maybe we'll take a small step back and just say, hey, pharmacokinetics. That's what PK is. Um, the way I think of it is, in our world, it's like, okay, before you give someone any sort of therapeutic, how can you measure their serum, plasma, tissue, CSF, whatever it might be, and measure the if there's any drug in there, which John, most therapeutics, it doesn't exist unless there's some form of it that you're trying to replace or put in there. But obviously, all small molecules, you shouldn't have anything.
Hmm.
Um, Unique monoclonal antibodies, likewise, you shouldn't have exposure. Although we're don't get me started on some of that. There's a lot of auto antibodies that might surprise us, but that we take these measurements and a baseline sample analysis is so critical to our world, right, John? That's
Yep.
or just finding out what the how sensitive can the method be in matrix, and that's always what it's about. And so for years, our organization, 44 of them to be precise, has been looking at trying to measure pharmacokinetic of small molecules, and then within the last 15 years it's been biologics, and now it's cell and gene therapies, and that's where it's gone from using reference standards and having LCMS parameters and LCMS criteria that was heavily dominating the field, and that kind of, for me, it's like really the late 70s all the way through the 90s, right, or halfway, maybe early 90s, middle part of 90s. That's when we first started. Oh wow, what are these murine monoclonal antibodies that suddenly we can produce? And if you remember, the cover at Time had the magic bullet, and it was all on monoclonal antibodies. I think it was like 92 that came out or something. And so then the field evolves, and John, it took a good 15 years before the 2001 paper comes out and they start really talking about biologics, right? And then
Yeah.
you fast forward to 2018 and 2019. The 2018 one, that guideline really starts to drill into just bioanalysis, and it starts talking about cell and gene therapies and really hammers home the differences between LCMS and biologics. And then I'll just say the immunogenicity piece, which really isn't relevant, but that's the 2019 document referring to. But that's, to me, a still big part of drug development. But with our focus on PK, I hope that's when I
Well,
say an overview of PK,
yeah,
that's sort of what I mean. Yeah,
I'm
you
going
can
to
go
have
ahead,
I'm
jump
going
in.
to
Yeah,
I'm going to have
add.
a quick counter to that. Yeah, often. Well, for years my kids never knew what I did, and I'm not sure they still do, to be honest, because we never really talked about it. But I do often have people say, "What do you do?" And I'll say, "Well, I'm an analytical chemist," and they'll say, "Okay, what's that?" And or I'm a bioanalytical chemist, and I'll say, "Well," and then so okay. And I would say we're working with pharmaceutical companies supporting their PK studies, and I said, "Well, how?" And I said, "Well, basically we're contracted. We'll be given a drug, be it large or small molecule. We'll develop a method to measure that drug in a biological fluid, usually blood, or plasma or whatever. But ultimately, for the PK, what we want to know is when a drug is administered, how high a concentration does it reach in the blood, and then how long does it hang around, i.e., how does it decay away? And obviously, for depending on the therapeutic need, some drugs you want to actually have a long cycle between administrations. Others you don't want to hang around very long at all. So part of what we're doing is showing that the drug that they're interested in developing, by measuring sequential blood samples, show that what the behavior of the drug is in the body, and that's basically pharmacokinetics. So that's usually how I talk about it, and keep the analytical piece out of it,
I talk about drug development, but that's a good, like, that's a good way to describe what we do, John. But interesting. And I even heard you say analytical, bioanalytical, right? Because that's an interesting chemistry, right? And
yeah,
I think of myself more in that biologist, immunologist state.
yeah,
Right?
yeah,
And,
yeah,
and you're right. It's all, but it still, all of my mind thinks of biochemistry, right? That
sure.
my
Yeah, see, from
my master degree is in biochemistry, and that's really set the stage for how I'm more on the immuno-oncology side, but I digress. So
I mean, that's where I come from. Basically, I came from chemistry, and my original degree had very little analytical, and it wasn't until I started doing analytical chemistry that I really found the piece I was comfortable with. And then as I got more into that, I didn't discover mass spec, but was
yeah,
introduced to mass spec, and having that detector on the end that could tell you what was actually coming out of a chromatographic column, that was fantastic for me, and that's why I've ended up doing, chromatography with mass spectrometry for the past 35 or so years
yeah,
now. So it's I came at it from a completely different direction, and obviously my career coincided with small molecule dominance, and as you're seeing this shift, not away from small molecules, because there's a huge part of the industry still focused on small molecules, but it's the rise of the large molecules, like you were talking about, the monoclonal antibodies, and then where we're talking about here, it's moving into more complex
yeah,
species that
yeah.
that actually take some of the properties of the MABs, combine it with small molecules, and as with ADCs, we then talking other therapies. And I'll throw it back to you, because
Yeah.
it's much more your space.
No, John, and it is and it isn't, right, because you touched on it. The modalities of drugs have gone. They're starting to blend together. They're like hybrids, is the word. But
Yep.
like
Yep.
antibody drug conjugates are now becoming. There's so many different iterations of those, right? And the one of the coolest ones out there that I've seen is where they have the antibody is now engineered in such a way that it does not bind. It won't bind unless it's in a certain microenvironment that cleaves off a peptide blocking the
Hmm.
binding region of it. That's so
Sure.
powerful to me,
Yeah.
John.
Yeah.
That
Yeah.
is so
Yeah.
powerful. And then on the other side of it, there's groups out there that dose them with ADCs, but they won't unleash their payload unless you dose it with another small molecule. So bioanalytically, the PK analysis for that gets really convoluted, doesn't it? Right? Like
Oh,
it's
sure. I
gone
mean,
from so
it
maybe a step
actually,
back, right? Maybe a step
yeah,
back. Just even the ADC, you had free drug, you had total, right? Free and total drug, that's payload, excuse me. Then you had total antibody, and oftentimes that might be good enough, but now there's the total ADC. So for PK alone, there was 4 methods. That's a major investment, John, right?
yeah,
And now you couple it with like different iterations of that, another small. It just gets a little like what is enough? Do I have to systematically break down each of them to do my specificity? Right? These are the types of things that are they relevant? Are they germane? I digress, Joe. Go ahead.
And one of the challenges here is now you have your so we're talk here we've come from two different directions. You're now forcing these two different directions to collaborate on
Yeah,
analysis to
yeah,
support the same study, because I think like a mass spec person, you think like a biologist, but we have to be together because or we have to work together because these species require the
Yeah.
expertise in both areas
And John,
to really give the answer that the client needs.
some of these ADCs that are highly effective, they have a lot of instability in matrix, don't they? And so
Hmm.
then you really need a lab that can sync up and test rapid, avoid freeze thaws, get multiple aliquots, be able to test them. But and that's just now we're just scratching the surface of it. If we think of now they're making, instead of like full ADCs, they're fragments of antibodies or other scaffolding that can be used to deliver small molecules, other ways to hone in to bind to the tumors. That's where, and they're making bigger, badder versions of all of it. It's fantastic, right? And then what's the biggest, most emerging version? It's the cell and gene therapy space, and that's to me, it's like that has to be its own. I almost prefer we give it a different term, John, instead of like PK, because when I hear the term PK, you think small molecule this, I think large molecule, right?
Well,
We kind of
actually,
have this, we have
we,
this
we
idea of what we need to do, but they I know we have to do some sort of studies, but to call it a pharmacokinetic is just unfair because there's no
sure.
way to track a cell like
That
that,
that's exactly that's exactly where I was coming from. At least I don't necessarily think of it as small molecule. I do think of it as small or large, and with those, you know what's being administered, so you know what you need to track. When you're administering a cell or a gene, what are you tracking? That's the challenge here.
Yeah. Even think of it this way: the drug product, what you get from the CDMO, we know what that's going to look like in our world. It's going to come with a certificate of testing, a certificate of
Hmm.
analysis, whatever you want. It's got all sorts of things to it. It's purity, this and that. You know, even something as simple as like a liquid nanoparticle. Sorry, I keep calling it liquid, an LNP, a
Lipid nanoparticle.
lipid nanoparticle, or oligos, or any CAR T cells, any sort of CRISPR technologies, and they're blending those all together. John, there's no reference standard really there. And you could say, well, Dom, they do have it, but the moment you put it into somebody, the recovery of that is way off. It's supposed to be incorporated. You may not see it. So then what becomes the default? Some of them, if I put 50,000 or say 5 million autologous cells into somebody, CAR T cells, whatever they might be, or even xeno cells, different allogenic cells, however they're coming from, I can't recover 50,000 of them, John. I can't. I'm lucky if I recover any, you
Sure.
know.
Yeah.
And so, what's that mean? And then, if you do, a lot of things are going right into the bone marrow. They're going into like certain, and then what my mind is racing. Right, it comes from, well, how many injections am I doing before I even think of, there's a standard regimen for small molecules and large molecules. We understand that they're going to do, okay, yeah, they're going to do some sort of multiple ascending dosages or whatever sort of SAD MAD that might be done, whatever that might be, right, John? We kind of have an understanding of that.
Sure. Yeah.
But in the gene therapy space, I mean, one of the biggest challenges is a single injection. Is it going to be multiple? They don't even like. How are you going to design your PK around that? So I feel like it's almost like got to be, and I often use like it's a PD PK, but I don't know. We've created the word immunogenicity and proteomics in my lifetime, John. I think we need a new word. You're just the guy to do it on this great Friday here in April, John. What word should we use for gene therapy PK?
I really don't know.
No, I'm throwing it back to you.
I hide when people start asking me those sort of questions.
No, but I don't, John.
I
That's
mean,
my rant about it. It's just the whole now there's certain things you can do. I don't want to make it sound like it's a black box, but there's biodistribution studies for certain gene therapies and cell therapies where you can try to find the microRNA on target, but then you have to do safety assessments of it off target. But none of it is. All of it becomes very difficult translating it. And here's the really hard part too: is non-clinically, I can do a lot of biodistribution, can I?
hmm
When I go into the clinic, it's a completely setup that's more. I'm more looking at it as like some form of viral count or viral shedding, because biodistribution to me implies a whole bunch of muscles, organs, whatever I need. If I go into a human, a biodistribution study is what? Fecal, urine,
yeah,
saliva,
yeah,
tears. Very different, right?
yeah,
Very different.
yep,
And so that's another, just layer it on, but not just the bioanalytical complexity of the methods. It's the whole way they're dosed, the matrix you need to look in. And, the good thing is, John, we solve a lot of these problems here.
So, from your perspective, is our biomarkers more relevant when you're
Yeah,
trying to
I
monitor
like the term
what a gene
pharmacodynamic though, but
Yeah,
yes,
yeah,
yeah, I
sure.
don't mean to talk over you. You're spot on. It's the pharmacodynamic piece of it, and then a lot of it. If my thought process, and I believe a lot of people follow this, it's the safety of it as well, because that
Sure.
tells you a lot
Yeah.
of
Yeah,
information.
absolutely.
Just
Yep.
just monitoring someone's T cells, for instance, for if their ability to generate a pneumococcal vaccine, for instance,
Hmm.
all sorts of ways to make sure you're not damaging the individual or hyper autoimmune.
Sure.
It's doing some
Yep.
sort of hyperimmune response, and that's that whole dual-edged sword that I said earlier today about, like, manipulating T cells is something I've been studying for 29 years now, John, and it's unbelievable how sophisticated we are today relative to what we were doing back in
Oh, sure.
1994,
That,
right? So,
that's
um, but
that's true across the board for everything
yeah,
we do.
and
Yep.
we kind of, so just to keep in with our agenda a little bit, we went over PK. We talked about some of the evolutions and then monitoring of PK, and I think we kind of touched on that, John. But that was kind of it for me on this particular thing. Oh, and then maybe the regulations, John. Unless you had something else to add,
So I
because
think
that is
that,
an
yeah. So
important
the
piece.
monitoring of PK, obviously I'm just going to step back to that. You got a challenge. So ultimately, then here's where pharmacodynamics and biomarkers that are being influenced by administration of a therapy, they become key. So the ability to identify a biomarker, and that's why we looked at articles like the Parkinson's piece, because looking at a biomarker that's there for the future, because the ability you might with these more complex therapies, you may lose the ability to monitor them directly, like we're used to with a small
Mm.
molecule or antibody. So ultimately, it's the it's the it's the it's the endogenous species that are either up regulated or down regulated, or impacted by the therapy. They become much more of a relevant
Yeah.
analyze. So you have to have the ability to figure out, how do we analyze these species when this complex therapy
Well, some
is
of it's
administered.
straightforward, right, John? If
Yeah.
I'm doing a
Yep.
rare disease and I'm using some sort of cell and gene therapy, and this individual, when I go to measure their level of a given enzyme is negative, and a normal patient that has 100
Sure. Yeah.
picogram of it, I do my treatment and they're up to 25. Well, that's going to show, to me,
Yep.
that's the PK
Absolutely.
measurement
Yep.
there, and
Yep.
to some
Yep.
degree, even though that that is scientifically not what it is at all.
Hmm.
But the bioanalytical method for measuring that pharmacodynamic protein, it's debatable. Certainly, on the surface, it should be validated as if it were a PK, but we're not making that. So now I'm using a commercial recombinant version of something, unless the sponsor invests in making it themselves. But either way, it's not the drug, John.
Sure. Yeah.
And then there's all
Yeah.
sorts of not philosophical. You often have to do a functional test of that protein just to be clear about it. So, say you're targeting a specific tissue, and the transgene is integrated, it will produce the protein, and you can measure that. Well, that's not enough. You got to see if it's functional, and more oftentimes a functional test is more germane. But that's the earlier the majority of the CAR T cells were in a space where that was easily measurable. Again, enzyme replacements or leukemia, lymphoma diseases, where you can just take cells out and look at them. As we get
Hmm.
into solid tumors and
Sure. Yeah. Yeah.
even, like, now treating type 2 diabetics with it, much easier to monitor some of that, because it's, insulin's the biomarker, right? So there's some established diseases where those types of pharmacodynamic tests are surrogates for the PK.
Sure. Yeah. Yeah.
But others, like in the immuno-oncology space, it gets trickier. Other than, like, sometimes it's like these people didn't respond at all to a pneumococcal vaccine, and suddenly they are. That could be, that to me says, oh, I've augmented the immune system in a positive fashion. Now,
Sure.
do
Yeah.
I have T cell specific responses against the tumor? Well, that's another flow test. But maybe you don't have that. But if heck suddenly these individuals lacked functional T helper cells at all, well, there's something there is going to be beneficial. But maybe I have some biasness in this space dating back to my earlier years. We didn't have tetramer technologies and some of the stuff that exists now, John. If you might not know what I mean, but you can monitor T cell receptors through things like tetramers and other things, so they mimic MHC complexes to say, okay, yeah, this T cell is specific for that, and that's how you do a lot of flow tests to measure it, and then an ELISpot test that didn't exist up until early 2000s or late 90s. And anyway, those are all things those are major PD assays for surrogates of cell and gene therapy.
Sure. Yeah. Yeah. So
Yeah, and maybe
now,
the last
now,
topic. Yeah, go
now I
ahead.
set you down that tangent. Now I think that then you can talk about the evolution
I
of
almost sound like
regulation.
it seemed like I had a brain for a minute, John. I don't know. I got into a trance like Will Ferrell does in um, Old School. No, I'm joking. So tell us the last topic here, and we'll wrap up.
Evolution of regulations to deal with these kind of species, and that's much more you're much more involved in that than
Yeah, it's
I
a
would be.
little wild west is how I kind of call it. And John, if you're we're blessed to have lived through the transition from small molecule to large molecule. So I think of it as where the
I'm
large
not letting
molecule.
those small molecules go, you know.
Yeah, no, but like where we were in like the early 2000s, in terms of like just the field was so dominated by small molecules, and
Mhm.
then the regs were like, oh, by the way, there's these biologics, and then so it's taken to be, it feels like a 20-year battle to get a separate document for, well, not separate, but at least a different mindset, a different
Sure.
guideline for the biologics. So we're kind of back in that first 5 to 10 years of these. There's a lot of CDMO. I don't want to make it sound like there's no guidelines. So there's a lot of like GMP guidelines and manufacturing guidelines that are good roadmaps for bioanalysis, John. So I feel like I'm in a negative state on things, but those regulatory documents are really important to help drive how we're going to get into the actual bioanalysis. And there are there's some, they escape me right now, but there are some PCR based. They're not like FDA guidelines, John. What we're really looking for, right? Like a
Sure. Yeah.
2018, you need to follow this. No M10 adjustment type guidelines. There's a lot of things that the industry prefers, but like most things, they start to alter, they start to change. The biggest challenge right now is practices, in my opinion, especially for emerging modalities. Less the ADCs, I think we're ironing all that out.
Sure.
It's more in that specific cell and gene therapies, the CAR T cells, the CRISPRs, the LNPs, anything that's delivering a viral message or a viral DNA or whatever it might be. Those types of things often are run through CLIA laboratories, and they have a different set of standards that they do versus a GLP lab. And I think that's probably its own podcast. But on the surface, there's a lot of practices. For instance, a CLIA laboratory can run a control or a standard once a day. That's it.
Yeah.
They'll run it once a day, or maybe even like gulp once a month. And if you really want to know some of the dirty details of a CLIA lab, they almost never fail a PCR experiment, John. Right? Like what world are we living in that might not happen to us, right?
Sure.
And
Yeah.
so I say that, but you'd hear, well, we got controls, this and that. I'm sure you do, but they're not anywhere near what we would do in a GLP regulated setting, because that's just not, we wouldn't be able to run. Let's call them calibrators, right? Standard
Mhm. Sure.
NQCs,
Yeah.
they go on every run, right? They go on every batch. They're not something we can do in the morning, and you might have some other practices there, but it's not the same. And then singlets, John, they do a lot of things in singlets. And then there's the emergence of ddPCR, which that's its own little challenge, because um, in theory, you're trying to quantitate each piece of message. In the
Yep.
ddPCR, it's how many cycles before I can see the message. So there's still, I think we've ironed out what the lower limits of quantitation are for qPCR, but the lower limits of quantitation for ddPCR are still, again, not what it's a whole nother show, but that's still emerging. So I think those are the major ones.
Yeah.
As for flow cytometry and ELISpot methods, I think they've got good understanding on how to develop and validate those methods. There's ample ways of using surrogate cells or surrogate. When I say surrogate, I mean healthy donor or
Sure. Yeah.
cell line or wherever it might be to control your experiments. Because that's on the surface, what are we talking about? For development of a PK PD marker or a PK assay or even some sort of surrogate PK, you're verifying the equipment, you're verifying the reagents, and then the verification of the instrumentation, right? I think I said those. There's three things: the individual, the reagents, and then the instrumentation. Because there's no reference standard, John, so it becomes a different thing.
Yep.
So that's my diatribe on it.
So like you say, it's at that stage where we were probably 30 years ago in LCMS, where it's really trying to find common ground for the
Yeah,
long
and
term.
the only advantage now is when small molecule came, the FDA was like, "What are we doing here?" Right?
Yeah.
Now there's been two other paths that have been carved that help.
Yes,
It's
exactly.
not, I think
Yeah.
it's got to be. It'll be a lot more. I don't want to say quicker or faster, but I think that's part of it. And I don't want to say it's like that there's confrontation with the FDA, but it seems a lot like more back. It's always been back and forth. It's just I think there is more reliance on the FDA is like, okay, help us. Here's our challenge, and then here, and that's just a cause. That is due to improvement in communication, even things like Teams and all sorts of information flowing, and even artificial intelligence to help us, John. So I hope that made sense where I was going there in terms of it should not be as hard to iron these things out and shouldn't take as long.
I would hope not. I mean, yeah, I know that like in the early days, there was people had firm ideas on so many different things, but they had to thrash them out to come to a sort of understanding. But
It's always funny to see scientists getting angry.
Yeah,
it's cute.
When I think of some of the contrasting acceptance criteria we used to have to work to, because we were quite often working to client SOPs rather than our own, and some of the things that some people would accept and others just wouldn't, and somehow you, like say, you got to get compromise on that. So yeah, hopefully people learn from that experience.
And I think they're better about trying to be clearer on what is a no and what is okay to do.
Yep. Yeah, exactly. Yes.
And even some of it, I don't want to hate on the CLIA labs too much. They're looking to engage our space. We're looking to engage them. I think it's a nice, in my experiences, it's been, because I do sit on like an AAPS subcommittee for PCR. It's nurturing, it's positive, but it's certain, some of it is definitely like, well, I don't think we can do a singlet, and someone else is like, well, why not? We've shown that it's reproducible, right? So that's
sure.
The conversation.
Yes.
But
I know, like at least in the LBA space, the EBF were really pushing on the syndicate analysis a few years ago, where it was more common to do the duplicate. And I it's as much as I know about it, but that was
I think it took a while to go from. For me, I had done triplicate all my life until I actually went to work at a CLIA laboratory, and then we did duplicate, and then I came here and realized, their duplicate is okay. Although there's still a fair amount of, not a fair amount. There are some methods that we use triplicate on, John. Still.
Sure. Okay.
I think that wraps it up for today, John. It's been fun. I don't know if we need to go over what we're doing with our weekend plans. That's up to you. I know we're short.
Me as well. While we're here, I'm not in any hurry to do anything. What's your plans?
Yeah, so a friend of mine, and I won't embarrass him. But you know who this individual is. He's been working hard over the last 6 months or not 6 months. I think it's been about 3 months. He's in a play called the Wizard of Pause. So this evening, my wife and my neighbor and friend the four of us are going to go watch our other friend. I'm being vague here, but again, I don't know if he'd want me to mention him, but he is a bioanalytical consultant, if that helps you. And so he's a fun guy, and he's got a role in this musical play, again, called The Wizard of Pause. Yes, there are dogs in this. It's based off The Wizard of Oz, and it's should be a fun night. And so that's kind of exciting. And then I go to APS. But John, I've had an incredibly busy couple of weeks. We had spring break. I think I mentioned that, but then I went and got to go visit my brother, and that was super exciting. I got to see a lot of family. I have a lot of nephews that are close to 30 years of age, and so I got to hang out with a whole plethora of Warinos. At one point, there was, I think, 11 of us, which was kind of cool, and that was enjoyable and always fun to spend 3 nights at 10,000 feet up in Cripple Creek, Colorado. It's a beautiful little part of the world, but John, you better make sure when you get there you have what you need, because it's there's no run into a convenience store, if you know what I mean. But that was I've been having fun there, and then I gotta talk about my big green egg, and actually I'm the Uni pizza oven I've been thinking more about that. I found a couple pizzas that I want to make on that, but my big green egg has been great to me, and really I've been knocking out wings with all sorts of. I'm really starting to hone in on the temperature on it, John.
Sure.
And so I'm getting wings that are like charred. I'm finding out ways. And then I made a tomahawk as part of because my nephew and I, we went out to Colorado together, and then he spent a few more nights here in Kansas City with me. And one night I made a tomahawk steak. It was this big, huge, look like a tomahawk. I don't know if you know what I'm talking about.
Yeah, another thing, absolutely.
Yeah, and I sous vide that and then seared it on the egg, and man, that was fun. So I proceeded to say, oh, maybe we shouldn't talk about this, and then I went on a long little trip about it. But I had fun. Your turn, John.
Yes, no. I mean, it's a relatively quiet weekend because, of course, I'm flying to California on Sunday, so that does sort of tend to dominate the weekend. We're in the sort of wedding planning mode with invitations have all gone out, and now we're starting to get a few back, and we're in active negotiation mode with relatives in the UK about trying to persuade them that they really want to come to upstate New York in September. And then this weekend is a record store day, so I have a list of things that I will be chasing tomorrow morning in Syracuse, and then there's a few in the UK that I'll try and order once I get home.
Fun.
So,
I said all that about me. My son's got his big prom this weekend. He's got his junior prom, so that's going to take up some time this weekend too. He's all excited about that.
Good for him.
Yeah. It's nice to be him. I'm jealous of him. No, but John, no, that's good. It's as always, it's a pleasure to do this with you, John. If you had anything else,
I think that's basically it. Last weekend we went, and my wife had a what's the word? An in on some kittens, and she's got an eye on a couple of those. So likely will be a multi-cat household again, beyond the 2 we currently have. But that'll be a couple of weeks off because of our travel plans. So
that's exciting.
Yeah, so there's things going on.
That's exciting.
And it's just, and right now it's like we're in that weird April weather where it's like hot one day and freezing the next, and it doesn't really know what it wants to do. Today's a hot day. We're in the 80s today. I think a couple of days time we're back down in the 40s.
Yeah. So John, within the same day that can happen, right?
Yeah.
And it's fun. It's lacrosse season too, so that's keeping me busy. And you'll usually they have a JV game and then a varsity game, and even though my son's on the varsity, we still go watch the JV, and because we do, and so you walk out of the house at 6, and it might be 75 degrees, and if you find yourself out at 9:30 at night out here, it might be 40 degrees. So the shorts you're in are no longer good, and that can get caught up. So I'm finally learning after 3 years of this, John. I figured out that I just need to wear pants.
you need to take a suitcase with you, right?
Yeah, no, it's funny. It just is. It's actually this year it's been great. Usually we have. April, you said it best. April is like, who knows what you're going to get.
Sure, exactly.
But no, glad to hear about the cat. And I think I told you offline a little bit about my cat woes, but we're optimistic about her.
Okay, good. Glad to hear it. Right, I think it's time to wind up. Thank you very much for this, and have a great weekend.